Ladies and gentlemen, the next event is the first plenary session of the morning. And the topic of the event will be vaccines and variants, which will be conducted in a virtual platform. And joining with us will be a well-known personality in the country in the fields of immunology and microbiology. And to introduce the speaker, I would like to invite Dr. Vaidehi Rajivan Francis, consultant microbiologist at the Teaching Hospital of Batticaloa.
Thank you, Lalisha, and good morning to you all. And it's my privilege and honor to introduce such an eminent person in Sri Lanka, Professor Neelika Malavige. And she's the head of the Department of Immunology and Molecular Medicine of University of Sri Jayawardenepura, and she's the director of Center of Dengue Research in University of Jayawardenepura. She's going to deliver a talk on vaccines and variants today. And she's the best person to talk about it. And she has pioneered all the work in COVID in Sri Lanka. And they have done the sequencing. They have pioneered the sequencing research and diagnostics in Sri Lanka. So over to you, madam. And we all are looking forward to listen from you. Thank you very much.
Thank you so much for the invitation. And to speak, I'm really sorry that I can't be there in person because I've only been to Battic twice, and I would have loved the opportunity to come, but because of time constraints, it was not possible this time. I hope everybody can see my slides. Okay. Now, what I'm going to talk about is where we are now, the threat, sorry, the spellings are wrong, threat of Omicron, and will vaccines end it? Because I think people want to hear about Omicron as well, about what is going on. Now, this is where we are right now. Southeast Asia, South Asia, and Africa are doing all right, but you can see that there are a little bit of cases surge in South Africa. Whereas we know that Europe was not doing well even before this Omicron news came. And also USA was not doing well because there was a large number of cases in these countries. And also Austria went into a lockdown. Romania had bodies piling. And why was this? Okay, I mean, because those appear to be the highly vaccinated countries. And when we look at the number of deaths, again, despite vaccination, when you compare Asia to Europe and America, Asia and Africa seem to be doing better. Now, that is because we know that there was a huge outbreak in India, and that although the vaccination rates are less in India and Indonesia, so many people got infected. So natural immunity in these areas also would have played a role in these differences in cases and deaths. And this is the vaccination, fully vaccinated population, and Canada, Australia, and China are doing well. And you can see that Sri Lanka is also better than USA. And this is despite, if you look at the proportion of fully vaccinated individuals, if I'm just talking about USA, it's between 50 to 60%, whereas Sri Lanka, it's more. Now, this is despite vaccines being administered to children, all children over 12, and they have also started vaccinating children younger than 12. The fact is, we know that severe disease occurs in old individuals, and about 1/3 of old individuals have not received vaccines in India. And even in Austria and Romania and certain other European countries where we know that, as I just showed you, the vaccine outbreak is raging. Although the vaccination rates are high, that includes children over 12 years, whereas you know that in our country, we haven't started less than 15 years yet. So, but the vulnerable, the older people, only 1/3 have not been vaccinated because of the very strong anti-vax movement and COVID denial. So there are people who in these countries who don't think, and even in Sri Lanka, who think that COVID doesn't exist, and it's a like a fake story, and there is no such thing as a SARS-CoV-2 virus, and of course vaccines are also fake. And because of that, you can see, despite the availability of vaccines, this is going on in these countries. And of course, these countries have also started giving booster doses, and European countries, UK are leading, along with Chile and certain countries. I mean, they use Sinovac, so they gave booster doses to quite a large proportion, including Middle East and US countries. Now, until this Omicron story came last week, all of us were, I think, quite happy. Because when you look at certain countries, things were, especially in Asia, things were looking pretty good. This is India, where it had a large surge. The number of cases were hitting a baseline. So basically, COVID was looking like it is becoming endemic, or is endemic in India. And the falling death rate followed, because we know that the deaths follow cases later. And this is South Africa. As you can see, there were hardly any cases in South Africa for 2 months. The deaths followed that drop. Same in Japan. Okay, cases were like hitting the baseline in Japan, and the same in countries like Indonesia. We know that in Indonesia also there was a huge Delta wave where there was lack of oxygen, bodies piling up. So that left a lot of people being infected. And actually, Professor Joseph Jacob from Velo, India, who is a very eminent professor, spoke with us last week, and he said that India, when you look at the seroprevalence rates and calculate, about 85.6% of the population seems to be immune through vaccination or through natural immunity. And because of that, and that is the proportion of people that you need to be immune to sort of make something like Delta endemic. So India, you know, is having a lot of functions. They have restricted everything, relaxed everything, and India is okay. That is because they have reached herd immunity through vaccination and through natural infection. So when with all this data early last week, we thought, okay, things are, you know, this is the end of it, and we are coming to the end of this pandemic. Because after all, we got Delta, which rapidly replaced all other variants of concern like Alpha, Beta, Gamma. And as of early last week, or even now, 99.5% of sequences sequenced everywhere in the world belong to Delta, which just other variants accounting for 0.5%. So because of its higher transmissibility. So this is just compares the transmissibility of Delta with other variants. This is the ancestral SARS-CoV-2. And of course, Delta is much, much more transmissible. And we know that it's the highly transmissible viruses that have a survival advantage, and it's the survival of the fittest. So Delta very rapidly displaced everything else. And from time to time, okay, there have been concerns about certain variants. There was concerns about epsilon, lambda, iota, and of course mu. This is because when you look at the parental or the ancestral SARS-CoV-2 variant, there was so much immune evasion by, for instance, mu, much more than the immune evasion by beta. And we know that even in Sri Lanka, there was like so many news reports about we are watching out for mu and so on. But so mu originated in the Latin American countries. So this is Ecuador, where they had iota, gamma, and then mu. But when Delta hit Ecuador, mu vanished. This is because Delta is more transmissible than mu. The same story in Colombia, where mu was accounting for 100% of cases from June to July. But then Delta hit Colombia, and you can see the cases are gradually declining. Same story in Chile, there was gamma, then lambda came and mu came, but then when Delta came, all the other variants vanished, and it is, you know, as you can see, Delta. So there was talk that, okay, it is Delta that is the last super variant. And when we look at the data in Sri Lanka, we have been sequencing the SARS-CoV-2 variants in Sri Lanka, and our sequencing has been quite good compared to other countries in our region. The WHO recommends sequencing 1% of the positives. we have not reached that level yet, but we are doing far better than the other Southeast Asian countries and South Asian countries in the proportion of viruses we sequence. And this is just our sequencing data from the beginning of the pandemic. I mean, which would be from November, because I mean, although we call that first wave that happened in March, April, that was not really a first wave, but just a collection of sporadic cases. So actually our first wave started in October, which was due to the ancestral variant and which was rapidly displaced by Alpha. And then when we got Delta, that was also rapidly displaced. And now we have different sublineages of Delta inside Sri Lanka. And so we have two Delta sublineages unique to Sri Lanka. So the so-called mother Delta in Sri Lanka gave birth to two unique Sri Lankan Delta sublineages. The first one called AY28, which we have managed to distribute to 14 countries. And this reflects where travelers from Sri Lanka go to. So this AY28 was a little bit of a concern because it had this particular mutation, which gave it a higher transmissible advantage, but it didn't seem to be very much threatening. And this other Delta Sri Lankan sublineage, AY104, again, we managed to export to several countries. We managed to export this to a larger number of countries. So we have been exporting quite well. But this AY104 is not, didn't have any mutations that were of concerning when you compare to the parent Delta variant. Now, all this was fine, and people were thinking, oh, it's just Delta and the sublineages, which were not very concerning. And so we are coming to the end of the pandemic. After all, Delta was identified last November, which is 1 year ago, and this is it. However, this happiness was short-lived because last week we know that the WHO within a few days announced that this new variant Omicron as a variant of concern. So this is the 5th variant of concern, so they didn't wait like they waited for the other observing the other variants. They immediately declared it a variant of concern because of the particular mutations that Omicron had, and it looked a little bit nasty. So as of now, as of this morning, these are the countries reporting Omicron, and it is so much widely reported in Europe. And now data is showing that it was actually detected in Europe before South Africa reported it. Actually, 2 weeks before South Africa reported it, it has now been detected in whole samples in Europe. So I think there's a high possibility it is there in Sri Lanka as well. We are currently sequencing today, but of course, I mean, we haven't been sequencing all around Sri Lanka very widely because it was just Delta. So there's a high chance that we could be having it, and other countries in our region could also be having Omicron, given that Europe and all these countries have it now. And the thing about Omicron is, so this is like a family tree. Okay, so when it's a family tree, it branches out like that. But if you see Omicron, the Omicron has suddenly appeared out of nowhere. And you don't get viruses suddenly appearing out of nowhere without any parents giving birth to this Omicron. So it's just like suddenly coming out of this very old parent, which is impossible. So there are several hypotheses about this, how Omicron suddenly appeared out of nowhere. One thing is because it's Africa, it could have been circulating for some time before it got detected, and people don't think so because it looks like it originated in October this year. The other thing is it could have been sort of in a human host, immunocompromised human, such as HIV patient, causing chronic infection, leading to so many mutations. The other hypothesis is that it was a reverse spillover effect. So we know that the SARS-CoV-2 virus, we got it from an animal, and we know that even with Denmark, like a good example, that we gave the SARS-CoV-2 virus to animals, and we know that even snow leopards and so many animals died because when they were infected with human SARS-CoV-2, but then the animals can give it back to us. So if the virus mutates in a different host and gives us back to us, then it can have very different mutations and a lot of mutations. And as you can see, when you compare Omicron, the number of mutations in Omicron with the other variants of concern, there are so many mutations in Omicron. We don't know what these mutations mean. So right now, a lot of things are unknown. But the initial data looks like it is possibly more transmissible than Delta. Again, we need more data. And this is the test positivity rate in Gauteng, which is a South African city, which reported this Omicron. So in the 45th week, the test positivity of this province was less than 1%, but the very next week, the test positivity went up to 30%. So this was a concern that it might be more transmissible, but again, we don't know. We'll have to wait and see. So now coming to vaccines, I think things that we really want to know, given the landscape of the variants we are seeing, and especially Omicron, will variants escape immunity? And about the boosters, will we need yearly boosters? Does everyone need boosters? Do immunity from all vaccines decline? And are there differences between different vaccines? Now, we had a golden opportunity to study this because we are the only country actually in the world which uses 5 different types of COVID vaccines. We have the AstraZeneca, Sinopharm, Pfizer, Moderna, and Sputnik. And Sputnik, again, there's a Sputnik Light marketed, which is just the first dose of Sputnik versus the 2-dose Sputnik. So I won't talk about all the different vaccine types and our studies, but I'll just highlight certain studies, certain data that is relevant, I think. And when you take the virus, it is a clown-like virus, and you develop antibodies to different proteins in the virus. You can have antibodies to the envelope protein, membrane protein, spike protein, but it's the antibodies to this binding site called the receptor type binding domain that prevent the binding of the virus to the ACE2. And those are the neutralizing antibodies. So just the presence of any antibody is not really helpful and doesn't indicate anything. It's the neutralizing antibodies that are important. And we've been looking at antibody responses to the receptor binding domain of alpha, beta, and delta, neutralizing antibody responses, total antibody responses, T-cell responses, and memory B and T-cell responses. I will not talk about all these different things we did. I will only concentrate on antibody responses because I think this is a very mixed audience and people understand antibodies a bit, but not other aspects of immunology. And the main reason why we looked at neutralizing antibodies is because neutralizing antibodies are highly predictive of protection. So those are the antibodies associated with protection. And if you just do some assay and get some antibodies positive, that doesn't mean much, because if the antibodies are to a different part of the virus, or even if it's a spike protein, so these AstraZeneca, Pfizer, and Moderna vaccines have the whole spike protein. And if you have antibodies to other regions of the spike protein, but not to the important region, again, that is not associated with protection. So we looked at neutralizing antibodies as well. And for that, we used this surrogate neutralizing antibody assay, which we had the opportunity of using in our own Sri Lankan patients last year because of our collaboration with Professor Linfaban. It was their laboratory that came up with this assay. Before it was out there in the market and commercialized, we had the opportunity, these assays were sent to us last April itself, 2020 April, for us to use this and work on it. So this assay is currently, this neutralizing assay is currently used in many of the vaccine studies published in very high impact journals. So we use the same assay. And for the Sinopharm and AstraZeneca studies, we recruited individuals from the Colombo Municipality Council. And for the Moderna and Sputnik studies, we recruited individuals from Kandy, because those were the areas that those vaccines were administered. And this is, I'm just going to show you Sinopharm and AstraZeneca. Because first of all, I think the healthcare workers who are listening in got AstraZeneca, and the vast majority of our population got Sinopharm. So I thought I'll be talking about that. And all the data I'm showing today is out there presented as preprint. So you can, anybody listening to this presentation can access all the data that I'm showing right now. So this is just showing the total antibodies to the receptor binding domain in individuals in different age groups. And as you can see, 3 months after 2 doses of AstraZeneca compared to 3 months after 2 doses of Sinopharm, in all groups of individuals, people who had AstraZeneca had significantly higher antibody titers. And all individuals who had received AstraZeneca were seropositive, whereas the seropositivity rates for Sinopharm were a bit different. I mean, in the young age groups, about 96.7% of individuals were seropositive, whereas the rates fell to 90% in those over 60. So these are not neutralizing antibodies, but any antibody. So 10% of individuals over 60 did not have any type of antibodies to SARS-CoV-2, which means that if 1 million individuals receive Sinopharm over 60, 100,000 of them are unlikely to have any antibodies to COVID. And this is why it would be important to take that booster dose. Anyway, coming to the direct comparison after 3 months. So these are the less than individuals less than 60 years of age, which is for AstraZeneca first dose, AstraZeneca first dose, second dose, and Sinopharm. So this is Sinopharm at 3 months from the second dose. This is 3 months after the first dose of AstraZeneca. This is 3 months after the second dose of AstraZeneca. So individuals with 3 months after the first dose of AstraZeneca also had higher antibody levels than 2 doses of Sinopharm. So 1 dose of AstraZeneca gave stronger immune responses than 2 doses of Sinopharm. And this again shows the seropositivity rate with 1 dose of AstraZeneca versus 2 doses of Sinopharm and 2 doses of AstraZeneca. And again, 1 dose of AstraZeneca gave better seroconversion rates than 2 doses of Sinopharm. And this is in the over 60 population. In the over 60 population, these are the neutralizing antibodies because you call them ACE2 blocking antibodies. And 61.9% of individuals who received both doses of Sinopharm did not have neutralizing antibodies, whereas the neutralizing antibodies were significantly higher in over 60s who received 2 doses of AstraZeneca at 3 months since the second dose. And apart from those, I mean, we wanted to look at antibodies to different variants, and we wanted to look at antibodies to alpha, beta, and delta. These are for AstraZeneca at 2 doses at 3 months. And you can see that the positivity rates are over 50%, except for alpha, beta, and delta in over 60s, which was around 47, and for delta, 41%. Now for Sinopharm, the positivity rates for variants was very much less. Even in the young age group, the positivity rate was less than 15%. And in all old age groups, the positivity rates were less than 10%. And we wanted to study the kinetics of the antibody responses to the receptor binding domain. So this is when individuals received the first dose of Sinopharm and the antibody levels went up. This is at 4 weeks when they received the second dose. When they received the second dose, the antibody levels went up, but it gradually declined with time. So these are the total antibodies and not neutralizing antibodies. This is for AstraZeneca baseline when they get 1 dose, and it continues to rise. And this is when they get the second dose. And at 12 weeks after the second dose, it is continuously, from this point, it goes up rather than coming down. This is for the total antibodies. Now, it's the neutralizing antibodies that are more important rather than the total antibodies, as I mentioned earlier. So this is the Sinopharm, people who got Sinopharm at baseline. When they're given 1 dose, the neutralizing antibodies rise, but none of them, it doesn't rise to give a positive response. it is only after the second dose that individuals get a positive neutralizing antibody response, which actually declines with time. For AstraZeneca, with one dose, everybody gets neutralizing antibody except for a few individuals. But of course, with one dose, the neutralizing antibodies decline with time. Here, the healthcare workers get a second dose, and then the immune responses, neutralizing antibody responses rise. And of course, then we know that healthcare workers were given a booster dose at 6 months. So this is a 3-month time period. So we do have blood samples at 6 months, and we will be studying the effect of the booster doses, not right now. So we are following longitudinal responses for all these vaccines in individuals. And because of these differences in responses that I showed you, the WHO has recommended that in all the individuals over 60 years of age, individuals who received inactivated vaccines like Sinovac or Sinopharm, they should be given a third dose as a part of the extended series. So it is not actually a booster dose. They're talking about a third dose. The booster dose is a different type. So additional dose between 3 to 6 months following the second dose. So what does all this mean? Basically, vaccine platforms induce different type of antibody and T-cell responses. I didn't show the T-cell responses because I think it would be important to keep time for questions. But we need real-world data. There is real-world data for many of the vaccines like AstraZeneca, Pfizer, Moderna. There is not much of real-world data for the inactivated vaccines. So that is really essential. And now, so many studies have shown that booster doses actually do boost up the immune responses. So, but then will boosters be effective in a year, in 2 years, in 3 years, 5 years? What happens? And what happens when we get Omicron? So, and I think it's important for us to realize it is when we get Omicron and not if. There is no if here, because by any chance, if Omicron is more transmissible than Delta, which looks like it is, or even has a similar transmissibility, even countries like Australia, New Zealand, which have the strictest border control, could not keep Delta out. So if we think that we could ever keep Omicron out, that is, I mean, you're not being very realistic. So we will get Omicron. So when will that happen? I mean, we could already have Omicron. I don't know, but it will be an eventuality. So now about Omicron, we still don't know anything, like what impact it will have, because after all, countries like ours and many other countries have had vaccination, good vaccination coverage. And also we've had boosters. We are one of the Asian countries which have boosters. Other Asian countries are not as lucky as us. And we've also had natural infection. So what impact will it have? Will it evade vaccine immunity? And does it matter if it evades vaccine immunity? If it's going to be mild? Is it going to be highly transmissible and mild? These things is too early to comment about these things. But I would also like to show you this figure. Now, just because something is mild, it doesn't necessarily mean it's okay. So this red line is a more virulent virus, okay, whereas this orange line is a mild virus. So although the more lethal strain initially causes more deaths, because the rapid transmissibility of the mild strain, because of the exponential rise, eventually more infectious strains result in more deaths. So this is the unfortunate reality. Now, of course, coming to Omicron, this is not a surprise. People knew that this Omicron is going to emerge. I mean, a variant was going to emerge. Nobody called it Omicron. And it was going to emerge from Africa. Because I just showed you the data from Africa, what it looked like. And in some countries, the proportion of people who receive just one dose is less than 5%. So there's a large population of unvaccinated people. And here in Sri Lanka, we are thinking of Omicron coming when quite a lot of people have received 3 doses. But imagine when variants are coming in when there are no vaccines. So it's not just the being fair, But by not sharing vaccines, you're allowing more deadly variants to emerge. And when such variants emerge, you yourself suffer. And and of course, Pfizer and Moderna are saying that, okay, they can come up with a new vaccine, a tailor-made new vaccine for Omicron. And if you think, if Pfizer or Moderna make a new vaccine for Omicron, that you will get it very soon, you have to understand that you have to be dreaming. I mean, all the rich countries will hold the vaccines and not share with us. So the important thing is that nobody is safe until everyone's safe. I would like to thank our collaborators, Professor Graham Mogg and Professor Alan Townsend from Oxford, our collaborators from the Colombo Municipality Council, Dr. Dinu Guruge, Dr. Ruwan Vijayamuni, and our collaborators from Kendi, Dr. Suranga Fernando, and all our funding agencies. And all this work was possible because of huge effort of a large team of individuals working all 7 days of the week and some days even in the night to do research, not just PCR. So thank you for the opportunity to speak to you, and I'm very happy to take questions. Thank you.
Thank you very much, Professor Neelika, for your excellent informative talk. And I can see one question already typed on the Q&A. Would you
yes,
able to...
Yeah. So the question is there a difference between the vaccines given as a booster and the routine vaccines, or they are the same? Yeah, so the vaccines which are being given as a booster is the Pfizer vaccine in Sri Lanka, and even in other countries, it's either Pfizer or Moderna, it's the mRNA vaccines, because they have found to be more immunogenic. And at least with the mix and match of the clinical trial data of Pfizer, Moderna with AstraZeneca, this mix and match of vaccines have shown to be more immunogenic. So it makes sense to boost with Pfizer, Moderna. So the vaccines which are currently being used as boosters is exactly the same vaccines that have been given to billions of people in the world right now. We know that Pfizer has been given to billions of people in the world, although it was not so much used in Sri Lanka because we didn't have the opportunity because so many other countries had already ordered vaccines. So there is no difference, and these vaccines have already been given to billions, not millions.
Thank you. Yes. Any other questions from the audience?
What do you mean by AstraZeneca Covishield, which is given to Sri Lankans, no?
Yeah, so AstraZeneca, I meant Covishield. I mean, both are interchangeable.
Yeah.
And
was there any difference? Anybody studied the antibody response between those who received the original AstraZeneca produced in the Western countries and the Covishield produced?
Yeah, so India has done a head-to-head trial and antibody responses. That data was shared by the NMRA, and I think I don't know if it's published, but they had looked at the direct comparison of antibody responses, T-cell responses, T-cell functionality between the AstraZeneca made, not only in Europe, I mean, the AstraZeneca is made in several Asian countries like Indonesia, Korea, and so on, and Thailand. So the AstraZeneca one and Covishield, there had been a direct-to-direct comparison, which the data I saw, and they look exactly 100% similar.
Any other questions from the audience?
I don't see much questions from the audience.
Yes. Okay. So in the absence of questions, then I think we can conclude the first plenary. And thank you very much, Professor Neelika, your valuable time and very valuable information and enlightenment. Thank you very much.
My pleasure. Thank you.
Thank you, Professor Neelika Malavige, and also Dr. Vaidehi Rajivan Francis for this insightful session, and also for their valiant efforts as microbiologists during the span of this pandemic.